Hello, everyone, and welcome to our online lecture series for plant biology. Today, we're going to be talking about some of the things that dictate plant growth, recovery, development, maturation, all of the signals a plant sends throughout its vascular system, which we've now spent the last several lectures discussing, and instead focusing on the hormones that move through the plant. Hormones, which you may be familiar with through your own life as a human, are any molecules that signal or stimulate an action. These hormones do so by interacting with cell surface receptors to fundamentally change gene expression in the cell. So they are an external signal that's going to tell that cell that it needs to do something different from what it has been doing. It needs to turn on a brand new set of genes, turn off something that was there before, and fundamentally change its metabolic profile. So it's got to do something different. Now, hormones are just some form of chemical. Come in all different sizes. They come from different precursors, but fundamentally they are made by plants. So oftentimes they do have a lot of the basic building blocks from photosynthesis. So we're repurposing things in order to turn them into these signaling molecules. There is one signaling molecule, one hormone, that is above the rest in plant development. There is one hormone that is so ubiquitous, meaning found everywhere, and so simple, this is it. I'll just use the abbreviation IAA, or indole acetic acid. Full name being, I guess I can't just name drop without writing it out. Indole 3 acetic acid. And it goes by the common name auxin. Now, auxin is incredibly common. You can even buy it at your local hardware store or plant store. You can buy it in a bottle in powdered form as rooting powder. And as we will discuss today, that's because a lot of it is trafficked towards the root apical meristem. It is known as the plant growth factor. So the equivalent of our human growth hormone. It is important in growth as well as in healing. We'll have some cool images to discuss that in a little bit. It was actually first recognized by Charles Darwin. Contrary to a lot of animal biologists who like to claim Darwin's work on finches as his capstone of his life, that he was doing so much in terms of animal biology, Darwin himself was an avid gardener and paid much more close attention to a lot of the evolutionary and growth patterns of plants. But indole acetic acid, or auxin, was first isolated in 1926. So we've known about it for close to 100 years now, which is pretty crazy. The molecule itself derives from the tryptophan pathway. So it's important to know where these hormones come from. Tryptophan is an amino acid, which fundamentally is a building block for proteins. And that means that history from being an amino acid has a very distinct impact on the trafficking of auxin. Means that it's water soluble, and means that it should be fairly readily available. Tryptophan is a common amino acid, so you're going to find a lot of it. Now, auxin is primarily made within young leaves, as well as seeds. So it's going to be sourced from our green materials or early developing materials, and it's going to help maintain apical dominance. So you're going to move it towards your apical meristems of your plant, primarily towards your roots. So if it's maintaining your apical dominance, and it's an important growth factor, what happens if we break the auxin pathway? What happens if we mutagenize a plant so that way it doesn't produce the correct levels of auxin? And that's the way in which we study a lot of these hormone mutants, study a lot of these hormones. We develop genetic lines that are mutants for the hormone. After we've identified that they're the correct mutant for that hormone, we then try to identify any phenotypic changes. Say, for example, we want to understand how auxin plays a role in wounding. We would take a look at a plant that's an auxin mutant for lower auxin expression or no auxin expression and wound it and see what happens. Or if we want to understand auxin as a growth factor, we might grow a wild type, meaning a non-mutant plant, something that you would find in the wild, and compare it to our mutant. And this is actually what you see in this panel right here. On the right side, we have an auxin mutant. And on the right, we have the wild type. Both of these were grown to complete flowering. Do you notice a difference between the size of the two plants? I think you certainly do, right? The auxin plant itself, and this is our lab rat of plants, Arabidopsis thaliana, or Theocress in the common, for its common name. This is a plant that's incredibly well studied, has tons of different mutants, and we can see that when we've mutagenized an auxin transport pathway, meaning changing the plant's ability or efficacy in transporting auxin to where it needs to go, the above ground biomass, or all the green material, is far reduced in the auxin mutant compared to the wild type. The flowers should be extending far, far up. But in the mutant, they're tiny. The size of the rosette, or the green material of the plant, is far, far reduced. So the overall growth of this plant, despite the fact that they've been given the exact same water, the exact same sunlight, the exact same temperature, genetically, this plant was unable to grow as much because it was an auxin mutant. Keep this in mind when we talk about agriculture. Plant genetics plays a huge role in how much productivity we can get in agriculture. And a lot of what we understand about plant growth is certainly connected to our hormones. Next to the diagram or the image of some research images on auxin mutants, we have a diagram of auxin's role in root development. We will see that auxin being made in the leaves is being transported down the vascular column acropedally, and this is a vocab term meaning that it's transported down through the xylem and transported towards the root apical meristem. Once it reaches the ram, it transitions to basipedal auxin transport. So the acropedal auxin transport, meaning upward from the base towards the growing tip, means that the auxin will move towards the ram through the vascular tissue. And then after it reaches the ram, it will have basipedal auxin transport, or backwards, back towards the base. And it will do so in not the vascular column, the adjacent tissues, oftentimes portions of the cortex. So you've got this funneling where the auxin will move in high concentrations down before kind of splashing back up on either side, meaning that the highest auxin concentration will be at that root apical meristem. And it is that extremely high concentration of auxin that helps maintain the shoot apical meristem or root apical meristem in the first place. This directionality helps dictate where the ram is in the first place, or the SAM. It helps indicate to the plant where the region of cell division, where the region of elongation, and where the region of maturation are. Concentrations of auxin will increase on your way to the region of cell division. There's one other thing about auxin's transport that matters a ton, and we'll see some different mutants associated with this type of transport of auxin. Auxin is said to undergo polar transport. Because it's moving through cells, it has to have a directionality. It just won't radiate from one cell to all of the neighbors in all different ways. Instead, auxin's transport is polar, meaning it has a side. It will enter a cell from one side. In the case of acropedal auxin transport, it's entering from the top. And then there are receptors, transport proteins, called pin proteins. That help direct the auxin from one side to the other, so that way they can go from one cell to the next. And they do so and change the concentration by essentially having more pin proteins on the side that they're traveling than the side that they're receiving. This means that you will always be trying to transport a lot more pin proteins in that direction, that acropedal auxin transport. Now, pin proteins don't actually stand for anything. It's the pin is not an acronym here. It has to do with the phenotype of the plant. Their flowers were very messed up when we mutated the pin proteins. They formed a pin shape. So they were given the name pin formed. And as it turns out, the evolution of these pin proteins corresponds, or rather the presence of these pin proteins corresponds with the first plants on planet Earth that grew via apical dominance. So these are very ancient proteins that are very, very essential for plant growth. As I mentioned before, they're also involved in wounding. And so, for example, wounding, like the cut that we see here, can interrupt the vascular structure of the plant. You can rip the leaf a bit, and where there was a connection, there will no longer be, and this will oftentimes lead to a lot of water loss in the plant. But you still need to somehow get water towards the tip. Auxin can help reconnect vascular structures. So it is the expression of auxin at the site of the wound that will start this redifferentiation, so that way tissues that used to be parenchymal may start to fit the bill of being water transport tissues. And now you can get a connection between the xylem, which is pretty crazy that you can reinitiate regions of maturation simply by the expression of this protein. Now, I personally have quite the attachment to these pin proteins and auxin as a hormone itself, because my wife spent a large portion of her career studying transport of pin proteins in the plasma membrane and studying this phenomenon of pin migration specifically, moving from one side to the other and changing concentrations so that way the concentration gradient of auxin will be the highest at the growing tips of the plant. We additionally, as I mentioned, see auxin expressed in seeds, and auxin expression in the seeds is very, very important. This image from your textbook has 3 pictures of 3 different strawberries. The first has its seeds still on the strawberry. A smaller strawberry, not necessarily one of those giant octoploids that you buy at the grocery store, but a strawberry nonetheless. And the presence of the seeds, which are secreting auxin, allow for proper development. In panel B, all of the seeds have been picked off. Now, with the seeds removed, the rest of the fruit doesn't develop. It withers away and is no longer edible. This is very distinctly caused by the lack of auxin. When we remove the seeds in a singular band around the fruit, we see normal fruit development up top, but at that band in particular, we see a decrease in the thickness of the fruit, meaning that it was not receiving all the resources that it should have been. And we see that even regions after that are also underdeveloped. So you can get real changes in how things are developing based on removing sources of auxin. Again, that's all connected to that pin protein transport. Now, auxin mutants and pin protein mutants can have a large impact on root development, as you might imagine. So I wanted to also include some images of how auxin mutants and auxin transport proteins, the pin proteins, there are a number of them, not just one, can change the phenotype of auxin growth. So we have in the top left a wild-type plant grown in Murashigian Skug or MS media, the basic plant growth media. So this is just the stuff that a plant will need to grow. And then we have a variation in some other additives. And we can see that stressed out plants, which that's what the bottom panel is trying to show, stressed out plants that are mutants can additionally get even worse phenotypes when it comes to auxin. And so you might expect an auxin mutant to affect growth, but then additionally when the plant is stressed, having that auxin mutation will make you even worse off. So in this way, stress and mutants can compound the problems. Auxin itself it can be synthesized, as suggested by the fact that we have auxin mutants, are actually a common form of pesticide. So let me say that again. Rather, not pesticide, herbicide, sorry. So analogs, so molecules similar to auxin, can be used as herbicides. Meaning that we can synthesize a molecule that's kind of like auxin, but a little bit different, that gets absorbed by a plant that we don't want growing in our fields, and it causes the absorbing plant to really change its growth pattern. In many cases, this overabundance of growth, like a huge amount of growth hormone dumped on the plant, will cause the plant to effectively kind of blow up. And this use of auxin analogs is actually our third most common herbicide. Oops. And of course, in a botany class, we will be talking about what are the types of molecules that we use in agriculture later in this class. But it's important to keep in mind that auxin and its synthetic analogs can be used for this purpose, can be used to target things that we don't want in our agricultural fields. So that's the basic schematic of auxin. Probably the most important plant hormone, and certainly one that you needed to keep track of in this class. The plant growth factor that's also used in healing, synthesized in young leaves and seeds, and transported acropedally to the tip of the plant. And then after reaching the tip of the plant, it reaches its highest concentration and then is transported basipedally back from whence it came, along the axes of the ram. Now next up, we have another similar molecule, this being cytokinin. Cytokinins are a class of molecule that are very diverse, have a basic blueprint that looks like this if you're chemistry inclined. Cool, that may mean something to you. If not, don't worry about it. But their name derives from the fact that they were discovered promoting mitosis. One of the important steps of mitosis is cytokinesis, which is the splitting of the cytoplasm, and it was this splitting of the cytoplasm that cytokinins were seen to induce. We know that they do a lot more than just that. They were discovered in the coconut. Which, as you can imagine, have to be performing a lot of mitosis or cell division to help make the fruit quite large quite quickly, and then ultimately produce the edible portions of the coconut as well. They are in high abundance in coconut water. They are primarily made at root tips. Which is in stark contrast to our auxins. The cytokinins are made in the root tips and then transported via xylem elsewhere in the plant. Xylem transport. So they are made at the source, promote cell division, and then get transported by the xylem elsewhere. But obviously they're going to be needed in their highest concentrations with those regions of cell division. And just like we saw in what's called a loss of function mutant, meaning a broken version of a cytokinin, we see that these broken cytokinins lead to much, much reduced plants. So, yeah, not a lot of growth, not a lot of cell division, not a lot of mitosis happening in a cytokinin mutant. But on the opposite side of loss of function, we additionally can see what happens with gain of function mutants, meaning overexpression or making more cytokinin than you would normally. And I include this image. You'll notice that I am including a lot of images from primary research in this lecture. You'll always find the citations associated with the image nearby. But from this paper studying gain-of-function mutants of cytokinin receptors, so these are things that can sense cytokinin, we see that there are changes in overall plant size. The wild-type plant is this big, whereas these mutants that can perceive cytokinins get much more bigger vegetatively. Which again, if you are aiming for a plant that's going to be bigger vegetatively, something that you eat the leaves of, like one of my personal favorites, spinach or kale, you may look into how different cytokinin mutants may help improve your yield. This again is done under the same exact conditions. So this plot right here has the same amount of resources, light, and water as this plot here, but the size of our cytokinin mutant or our cytokinin receptor mutant with overexpression is just bigger. So again, a way that we can improve crop yields without changing their environment. Keep all those things in mind when we're talking about agriculture. So that's the basics of cytokinin. And we'll continue to go through our list of hormones and kind of summarize where they're made, what they do, and take a look at some of the mutants and variation that we see. Next up, we have another common hormone called gibberellin. Gibberellin is made oftentimes in young tissues. And is meaning that it's made in the young stem when the plant is growing. It is involved in hyper elongation, meaning that you will get variation in the internode size with the presence of gibberellin or higher expression levels of gibberellin. To understand this, we can take a look at these three plants. The first has a very short internode length, emblematic of what the plant might look like as a gibberellin mutant. The length between the leaves is very short. Flowers may develop properly, other features may develop just fine, but ultimately the distance between nodes, or the internode length, is very reduced in plant 1. In plant 2, you have a medium internode length, and of course, in plant 3, you have a longer internode length. And so if, for some reason, plant height was exactly what you're looking for, you may want to express gibberellin at higher levels. So if you're a plant breeder trying to make a very specific height sunflower, maybe you don't want it to be too tall to fall over, or too short to be boring, you may be screening your mutants or your plants for variation in levels of gibberellin. Kind of cool, right? So if gibberellins are responsible for hyperelongation in stems, you might also ask, what the heck is up with these grapes? These are Thompson seedless grapes, so the white grapes that you might buy at your grocery store. And you can see that the cluster on the left has smaller grapes that are more bunched up, overall producing less weight, whereas the cluster on the right has much larger grapes. You'll also notice that the stems are a little bit longer. The distance between the grapes is a little bit more on the right. The cluster on the right has been treated with gibberellins. The cluster on the left has not. And so, in this way, you can actually treat your crops with hormones during something like fruit development and increase your yield. So timing this and timing the application of hormones, not pesticides, hormones, can also improve crop productivity. Now, the way in which gibberellins are causing this hyperelongation of the stem is that in young tissues, they will stimulate cell division, causing that internode length or the distance, drew that in black, not going to be very useful, the distance between things like our grape clusters to increase. They're also causing the number of cells in our grapes to increase by stimulating cell division. So you're literally getting bigger fruit as well, all through hormone treatments. Now, next up on our list of hormones is one that you probably have some firsthand experience with. It's ethylene. Ethylene is ethylene, there we go, is something that's very commonly used in, has many different commercial applications. It is a hormone that is made in most tissues. It is a response to stress. I write that a little neater. It is a stress response hormone, and it's also very critical in a fruit process and a plant process. In fruit, it will cause ripening. And in the shoot, or in old leaves, old shoots, it will cause a process called senescence, which is, planned dying. So the ending of the life cycle, the programmed pulling back of nutrients, something that you would see in deciduous plants that want to pull away their nutrients from their leaves in the fall time. In fruits, like in bananas, which are giant factories for ethylene, we see them cause very rapid ripening. And so what's one way in which you can increase the speed at which your bananas ripen? You can place them in a brown paper bag. This is because ethylene is aerosolized. Unlike being transported in the xylem or cell to cell, as some of our other hormones, ethylene can be released into the air and perceived from the air, which is pretty unique. We know that ethylene, as its precursor, uses the amino acid methionine. Hate that button. Gets me so commonly. Sorry, y'all. And we know that dosage of ethylene matters. So let's take a look at this image from your book. This shows what a plant might look like under normal conditions, normal ambient ethylene. And then we see a curvature response in dark grown peas, where the more ethylene you'll get, the more curved and shorter your stem is. You change in a lot of different ways in a dosage dependent fashion. And so one of the things that we try to do is really regulate how much ethylene is given to our fruits in grocery stores. You can also pull ethylene out of the air in some of those green produce bags. They are lined with things that will bind ethylene and pull it out of the air, meaning that there's less of it penetrating into plant tissues, and therefore slowing down the ripening process of your fruit, keeping it from becoming spoiled quicker, or even in some of your vegetables, the stuff made of stems, slowing down the process of senescence. Crazy, right? Next up, we've got the hormone known as ABA, or abscisic acid. We've talked about this hormone before in the context of water stress. So we know that the production of abscisic acid can close stomata. So it is made in mature leaves in order to close the stomata. It is exported from the leaves elsewhere in the plants. So that's their ultimate source, and then it can move around the plant to help deal with other factors in plant stress. But it's not just involved in closing the stomata. It can also suppress germination. And now, why would you want to suppress germination? Well, this corn cob gives you a great illustration of why. In this corn plant, which is a known mutant for abscisic acid, some of the corn seedlings are not producing enough abscisic acid to help prevent germination, and they are beginning to germinate while still attached to the cob. They're not placed in the dirt, they're not in ideal conditions. This corn is so ready to go that it's going to try to become a plant while still attached to the cob. And so regulation of abscisic acid is very important in terms of making sure that you're timing your germination correctly. Next up, we've got salicylic acid. A hormone that may be better known to you as aspirin. That's its trade name in terms of human pharmaceuticals. It plays a giant role in something called systemic acquired resistance. Because plants have immune systems as well. Systemic acquired resistance, oftentimes abbreviated as SAR, is comparable to our learned immune system in humans, but very, very different in its mode of action. And salicylic acid helps as an antimicrobial. So we use it to help, regulate heart rate and other beneficial side effects. But in the plant, it serves to kill small microbes and helps in pathogen signaling. So in letting the plant know that, hey, guess what? I got something that shouldn't be here, and we should do something about it. It was discovered in white willow, or Salix alba, hence the name salicylic acid. It's also important as part of its antimicrobial and pathogen signaling function. It is important for ion uptake, so helping plants get the minerals that they need, and in photosynthesis. But those are both secondary roles to its role in systemic acquired resistance, the plant defense mechanism, the plant immune system, comparable to our learned immunity. That's all we'll ask y'all to know about that. If they mean something to you, cool. If not, that's fine as well. Now, next up on our long list of plant hormones, we've got brassinosteroids. An incredibly diverse group of hormones that have lots of functions that are highly variable. So it's kind of hard to talk about something that can do so many different things. But one thing that they do very commonly is help the plant respond to gravitropism, or its ability to detect which way is down, as well as regulate xylem differentiation. So here we see an image from your textbook of cells before and after they've started to differentiate into xylem tracheary elements. So we start seeing their cell walls doing totally different things on the right. There's a lot of staining going on here, so you see the chloroplasts in green on the left and the chloroplasts in red on the right, but you don't see any differences in the cell wall on the left. These are protoplasts. They don't have cell walls as of yet. But after the expression of different brassinosteroids, they start developing these beautiful ringed cell walls around the cells because they're differentiating into tracheids. And brassinosteroids are commonly associated with that and the region of maturation. Now next up, we've got one of my personal favorite hormones, jasmonic acid. Also called jasmonate. It is a linoleic acid precursor. Which you may be familiar with the texture of linoleic acid because it is used in producing linoleum, which derives from linseeds, a plant. And jasmonic acid is again involved in systemic acquired resistance, but also in wounding response for the plant. So when a plant is wounded, it will begin to make a lot more jasmonic acid, and that actually causes the plant to reduce the size of the organs that it's growing. If you continually wound a plant, it will continue to make more and more jasmonic acid and continue to make smaller and smaller organs, resulting in what you'd call a bonsai plant. So if you have ever bonsai'd something or have seen the beauty that is the art of bonsai, you've witnessed the phenotypic effects of a lot of jasmonic acid in stunting the growth, miniaturizing the growth of what would be a giant tree to be a very adorable tabletop decoration. Now, jasmonic acid isn't only involved in this wounding response, it's also necessary in tuber formation. So it has a different role in roots. And it's involved in abiotic stress as well, meaning stresses that are associated with mineral deficiencies or water. We're almost to our end point of our hormones. 2 more to go. The first is one that I got familiar with during my master's program, a group of hormones called keratin. And keratins are made from burning plant material. So you might ask, what the heck purpose would burnt plant material have in terms of being a signaling molecule? These keratins are not made by the plant, but are perceived by particularly seeds. Because if the seed is going to detect the fact that a fire has just come by, it might be a great time to instigate germination. Because everything around you has just burned. There's no ground cover. You don't have to compete for sun as a resource. The sun is literally yours. So these plants, like Rhamnia coulteri, the bush poppy, a plant that I studied during my master's thesis, and tried to germinate. I germinated something, or tried to germinate something like 1,000 seeds in one batch of 1,000, where I germinated them in gibberellin and water. I got less than 1% germination. In another batch, I added liquid smoke, which contains carrageenans. And in that batch, I got greater than 95% germination. So it's pretty cool to watch how this plant, even though I was giving it the correct water, the correct soil, the correct environment for germination, because it didn't detect burning, it did not germinate. Now, our next and last hormone are called strigolactones. Oops. And these hormones are, again, not something that's meant for the plant itself, but are exuded by the roots. The reason why the roots are producing them and secreting it into the soil is that they want to instigate the formation of mycorrhizal fungi. Arbuscular mycorrhizal fungi. I have to say mycorrhizal fungi are that word is one of my least favorite things to spell on account of the fact that I think I'll spell it differently every single time if I'm writing by hand. Strigolactones are super cool in that they are one of the ways in which plants are trying to recruit mycorrhizae, recruit help, but they also help the plant deal with salt tolerance. So if the soil is particularly salty, you might see more arbuscular mycorrhizal fungi as helping the plant do a better job of gaining water, while also, on the plant side of things, having a role of suppressing branching. You get less branching if strigolactones are being made. Now, for my image of strigolactones, I included this very pretty wild legume. With its name being Lotus japonicus, not a lotus itself, just happens to have the same name. I actually don't know why they would decide to name the genus that, but it is one of the model organisms for studying mycorrhizal fungi and root relationships. Beans, as you know, are plants that are rich in nitrogen, and so they have an additional symbiotic relationship with bacteria that are anaerobic in their nodules that allow them to fix nitrogen. This ability to fix nitrogen means that a lot of bean plants, legume plants, are high in protein because protein is an essential building block, or nitrogen is an essential building block in proteins. So, this relationship between arbuscular mycorrhizal fungi and plants is very well studied in the beans. And strigolactones, some of the first studies on strigolactones were done in these legume plants. But since then, we've seen that not only do legumes make strigolactones, but we know that a lot of our cereal crops like sorghum and maize also use strigolactones to help recruit fungi that will help them grow in different environments. Again, a symbiosis that can help improve crop production. Now, in all of this, I want y'all to know that dosage matters. With all of our hormones, whether we're talking about our polar transport of auxin that keeps increasing in concentration towards our tip, or whether we're talking about cytokinins that are made at the tip and then transported away, the tip being the region of cell division, or gibberellins or ethylenes, which their concentration certainly matters in terms of how fast your fruit's going to ripen. Side note here, if you have rock hard avocados and would like them to ripen very quickly, say for a Super Bowl in the near future, you may want to put them in a paper bag with bananas, because those avocados will pick up what the bananas are sending, the ethylene that is, and ripen just in time for your game day. And this principle of dosage mattering is fundamental to the way in which hormones work. So that's where we'll end our discussion of hormones today, but I want to wrap in another topic because we've talked about some of the precursors for these molecules. I will just end by talking about another group of molecules that are made by plants that, instead of signaling their change, can sometimes be used to signal changes in us. These molecules are called secondary metabolites. A secondary metabolite is any organic molecule that is not directly involved in keeping the plant alive, meaning doing photosynthesis, meaning transporting water, meaning performing cellular respiration. These things help assist in living, meaning help protect the plant, but are not directly involved or necessary to keep it alive. So you can knock them out, the plant will live just fine, but it might respond to stress a little bit worse. These secondary metabolites, and the two categories of these secondary metabolites that we'll talk about today, are frequently alkaloids. And you will be very familiar in the pesticide role of some very common alkaloids. So just like we talked about some analogs of auxin being used as pesticides, that's a growth hormone that's necessary for plant growth, as we saw with the mutants. These secondary metabolites, these alkaloids, help a plant defend itself as it's being eaten. They attack the herbivore that's eating the plant. Two very well recognized alkaloid pesticides are caffeine and ephedrine. I love both. These two alkaloids are stimulants when ingested by us humans. Ephedrine derives from the plant ephedra and is used to help increase the heart rate. Ephedrine is an analog for adrenaline. So when you drink the tea made from the ephedra plant, you are drinking in an analog of adrenaline. So it has a lot of the same impacts. It's going to increase your heart rate. It is an appetite suppressant, so that's sometimes why folks would drink it as a means of keeping the appetite down if you're not going to be able to eat in the very near future. Suppressing hunger during times of famine, pretty useful. Caffeine does much the same thing, with caffeine also being an appetite suppressant and a heart rate increaser. Now, both of these are made by plants to help poison very small organisms. But you and I are not very small organisms. When we take small amounts of ephedrine or caffeine relative to our overall body size, we get a slight stimulant effect. Whereas if something very small were to take in a similar dose, its heart might explode. Very different and very effective as the pesticide. Now, in preparing this lecture, I started to read about what's called the biosynthesis of caffeine. How does a plant actually make caffeine? What are the precursors from caffeine? And much like heart exploding from too much caffeine, my mind exploded when I learned where caffeine originates from. Caffeine is just a couple steps removed from a single step removed from theobromine, which is a bitter compound in chocolate. And then it's just a couple steps removed from a molecule called xanthocene, which is one step removed from a molecule that you might recognize, guanine. This is one of your nucleic acids. In your DNA, you have A, T, G, and C. This is G. So caffeine is related to or synthesized from guanine. Kind of crazy to see where this is coming from. So alkaloids are commonly used as pesticides in plants, secondary metabolites. And the other secondary metabolite that you will be very familiar with, particularly at this beautiful falls in Michigan, are tannins. Molecules that are used for protection against herbivores and also stain water brown. What tannins will do is that they are quite giant molecules that are designed to bind to digestive proteins. So they gum up your ability to digest the plant while you're eating it. If you have a whole bunch of proteins that are gummed up, you then need to process them. And you won't be doing any digesting. And therefore your digestion is slower, and your stomach might begin to get upset. These tannins are found in everything from soil associated with very wooded areas. Tannins leach out from roots. But tannins are also found in things that carry caffeine, like coffee. You might be familiar with the concept of putting milk in your coffee. It changes the color from the dark color of coffee to maybe a lighter brown. What's happening when the milk is being added to the coffee is that the milk is full of proteins, and the tannins are binding to the milk proteins. When they bind to the milk proteins, they won't bind to the proteins in your stomach, your digestive enzymes. This enables your stomach to continue to digest things without upsetting it. And in this way, a lot of humans add milk to their coffee in order to their black coffee in order to reduce the likelihood that the coffee will upset their stomach. And so we've come full circle in discussing a lot of our plant hormones and some secondary metabolites, some things associated in other pathways of helping support plant growth. We will continue to expand and look at the diversity of secondary metabolite biosynthesis later on in this class. As a teaser for it, caffeine did not just evolve once in plants, but it is a molecule that's evolved several times in different plant families. And so, as foreshadowing for what the second half of the class will be looking at, the evolution of plants, we now understand what caffeine is, an alkaloid that helps plants in its pesticide action. We'll later be looking at where does it come from, not biosynthetically, but evolutionarily. I'll see y'all next time for a further exploration of some growth habits in plants.